Welcome to Lesson 3C on Punnett squares and non-Mendelian inheritance. That's a big mouthful of words, but I think you'll really understand it at the end. So let's start with what is a Punnett square. You may have even heard of these, you may have seen them before, but the simple answer is it is a graphic organizer that's used to predict all the possible offspring that some parents could have. Here's a simple example. Let's say we were cross-pollinating this green pea and this yellow pea, and we knew their genotypes. We knew that this one was homozygous recessive and this one was homozygous dominant. What are the possibilities? It might not be obvious right away, but you might be able to see what are the possible combinations. But here's how you would set up the Punnett square. You take a 2x2 grid, and you take the alleles from one parent and put it on one side of the square, the alleles from the other parent on the top of the square, and it doesn't matter which one is which, where you put them doesn't matter. You could have put these capital A's down here and the little a's up here. Then what you do is you take one letter from each side and combine them and see what genotype would that be and then what phenotype would that look like. And as you see in this example, it's pretty obvious. They're all going to have the exact same genotype and the phenotype. It was because this is homozygous dominant and this one was homozygous recessive. If this one was heterozygous dominant, it'd be a little more interesting. But this is just a very simple explanation of how to use a Punnett square. They get much more complicated. We'll see a few throughout this note-taking method. Now, who invented it? Of course, some guy named Punnett. Reginald Crundle Punnett, British geneticist. He was really famous in his study of genetics in the 1900s. And he is still remembered to this day as the creator of the Punnett square. It's something that's been used for now over 100 years. That's how useful it has been. Pretty cool that his legacy is still going on. Now, let's talk a little bit about probability. I know you've learned this in math class, but what the definition is, it's the mathematical chance that something will happen. And if you flip a coin, what are the chances that you will get heads or tails? And everybody always says it's 50-50, right? Only 50% chance of heads, 50% chance of tails. Technically, yes, that's correct, theoretically. And the way you would write that is you can write it as a fraction. You can say 1 out of 2 flips would be heads, or you could say there's a 0.5 probability of getting tails. You could say there's a 50% chance of getting heads or tails. Now that's only if you're flipping one coin. Now what if you're flipping multiple coins? And if you're flipping 2 pennies, what are the chances that you would get double heads or double tails or head and tail? Well, that we could use a Punnett square to figure this one out. And the 2 possibilities, heads and tails, both go on both sides of the Punnett square. So you could get 2 heads. And that's a 1 in 4 chance to get head. You could get head and tail, which is a 1 in 4 chance from this side, but also you could get it from this side, heads and tails. Another 1 in 4 chance, which is a 50% chance of getting one head and 1 tail. And of course, you can probably see that you could get 2 tails, which is one other 25% chance of getting that option. It's simple explanation. It's a much more relatable, I think. Instead of talking about alleles and genes and traits, we can talk about pennies too, because it's all about just what are the possible combinations of these things. So let's apply it to some pea plants, and we'll do our pea plant Punnett probability problem. Say that 5 times fast. Now, let's say we have a pea plant that has purple flowers, heterozygous, big P, little p, and they cross-pollinate with a purple flower that also is the same genotype, big P, little p. What are the chances that it could have white flowers? You might be able to figure this out in your head, but if you couldn't, you would use the Punnett square. So you take the big P, little p, big P, little p, and let's start crossing. Let's see, you could get a homozygous, big P, dominant purple flower. You could get big P, little p, where is a 25% chance of that heterozygous genotype, and another 25% means that there's a 50-50 chance that it will be purple but heterozygous. And you guessed it, there's a 25% chance that it would be homozygous recessive white flowers. So that's the basics of what is a Punnett square. Now, we will do some practice with them throughout the next level, and I think you'll really enjoy it because, like I said, some people kind of think they're fun. They kind of sort of feel like you're doing like a Sudoku or some kind of a game, trying to guess all the possible combinations of things. But we're going to switch gears now and talk about something called non-Mendelian inheritance. You could probably guess what non-Mendelian means. It basically means not Gregor Mendel. And that's because, not to diss him, it's just to say that he studied two alleles, white or purple, yellow or green. And they only had options of dominant and recessive. Well, it's not that simple in a lot of other organisms, and especially humans. And as you see in this picture here, if you know your cat breeds, you might see that this cat looks like it has a little bit of tabby and then a little bit of some other types of cat mixed in. And you're seeing multiple traits, multiple patterns on one animal. Pretty interesting. So let's talk about what is non-Mendelian. Here's a couple examples. There's one type that's called incomplete dominance, and that is when you basically get a third phenotype. When you cross some red flowers and some white flowers, as you see the genotype here, big R, and big W, this would mean red, this would mean white, and when you get a big R, big W combination, you get pink. Which we didn't see with pea plants. We didn't see that with the pea, purple or white pea flowers. So this is just called incomplete because you kind of see red and you kind of see white. So what's the human equivalent? Hair type is a common example that's used. So if a mom has very straight hair and the dad has very curly hair, then the child could have wavy hair. Straight, basically wavy means straight and curly. It also applies, I don't have a picture for this, but it also applies to height. If some 7-foot basketball player had married a 5-foot gymnast, their child will be somewhere in between there. Maybe 5'6", maybe 6'3", but the height is another example of incomplete dominance. Here's another one, codominance. So with codominance, the alleles for both are visible at the same time, but it's a little different from what we just saw in incomplete dominance. We got a new color called pink, but this time there's an example here with chickens. A black chicken and a white rooster can make what they call a checkered chicken. And those are the options, especially if they're two homozygous color combinations. But the checkered chicken, if you lean in a little bit, it literally does look like it's black and white, polka dots or checks. And that's just becomes a different breed of chicken. What's the human example of this? Blood type. So a dad who has blood type A is written, the genotype for the blood type is actually written AO, and the mom has blood type B, which is the genotype is written BO. So when they reproduce, it could be the dad's A and the mom's O, and then the offspring would be blood type A. It could be the dad's A and the mom's B, and now we have AB. Basically, they're both. This child is both A and B at the same time. Or you see here that it could get mom's B and dad's O, and that would be blood type B. Or they could both pass on the O, and now we have blood type O, which you can guess is a recessive one because it only shows up if O is the recessive half of BO and the recessive half of AO. So blood type is a great example for codominance in humans. Next, there can be something called polygenic traits, and that is when the traits, there's more than one trait that affects the traits are affected by more than one gene. The non-human example here is in seeds of wheat plants, the husk of wheat plants. There's 4 different 4 different alleles here, and 4 different ones here, and when you mix them you can get like kind of like a halfway in between color. And then if you mix them again, you get like all these possibilities, this huge range of possibilities from dark red all the way to white and some pink and some orange, because look how many different alleles there are for this one phenotype, basically. What's the human version? Skin color. And as you see, there's a lot of different genes. There's 3 sets of genes that determine skin color, from very light, fair skin to very dark skin. And everyone in between, it has just different combinations of either the light little d alleles or the dark big D alleles, dominant and recessives. And as you see here how they spread out, it does look a lot like the different colored of wheat husks that we see in the wheat plant. Then there's multiple alleles. So there can be 3 alleles or more that can determine one single phenotype, one single trait. And in the example with rabbits, they have these alleles combinations, the combination capital C, and then there's little c with a CH subscript, or just a C with a little H subscript, or just a plain old little c. So if they're capital C, which is dominant over everything, if it's present at all, then the rabbit will be brown. Even if it doesn't matter what other ones it's paired with, those others are all recessive. But then the little c with superscript CH, if that's present, then it would be dominant over the other recessive trait, which is just little c with the H superscript. And this CH one is called the chinchilla variety. And then this one, which has a little bit of white but black legs and black ears, they call it a Himalayan, is dominant over the other 3rd recessive type, which is albino. And only one possibility for having the albino rabbit is if it gets both little c's without the superscripts. Whew, that's a lot of letters. Now, what's the human example? Eye color. Now, yeah, we've talked about blue eyes being dominant over brown eyes. I'm sorry, the other way around. Brown eyes dominant over blue eyes. But it's way more complicated than that. There are green eyes, there are hazel eyes, there are even people who kind of seem to have purple eyes. There's even different types of blue. There's icy blue, and then there's dark blue. And so all I'm going to say is that it all depends on the combination of all these different alleles that you can see here. There's not just B's, there's A's. There's A's and big A, little a, big B, little b. And what variety you get depends on that combination of alleles. All these different color possibilities. Now, here's a little joke. I'm going to let you try and figure out what type of dominance are we seeing in this joke poster here. I'll give you a little bit of a hint. You see that the dad has horizontal stripes and the mom has vertical stripes, and the child has both. So that should give you a bit of a clue. Put that in your notes and see if you get the joke what type of dominance this is. Now, the last thing I'm going to show you is a video, but I'm going to make this a separate embedded video in Canvas because I don't want this video to be too long. This has been a favorite of students that I've showed this to in the past, and in fact one year it was so popular that it ended up being played at a bar mitzvah, and the kids were all dancing to it and thought it was great. But it's made by that Science Rap Academy. I've showed you videos of theirs before. Watch it and answer this one question. The boy has blue eyes and his parents both have brown. What explains that? And enjoy the music while you're watching. It's really good.